Okay, students. So I discussed about the first concept, which is people used in the screening of recombinant host cells, that is the transformed host cells with the recombinant DNA. So now we'll get into the second aspects which people use it in the screening process. That is using nucleic acid hybridization method. People go for screening of the particular host cells. So it's a very powerful method for screening. People use it for screening of cDNA libraries, genomic DNA libraries, or even the gene libraries. People use it. So here, very important requirement is going to be a probe. That is a nucleic acid sequence, oligonucleotide sequence. So which can hybridize or which can form, which is going to be complementary to a particular known sequence. So by using a suitable probe, we can able to screen the existence or the presence of a particular uh, sequence of nucleotides. So probes a DNA sequence that is used to detect the presence of a complementary sequence by hybridization with a nucleic acid sample. There are three main types of probes: cDNA probes, DNA probes, oligonucleotide probes. There is even people have RNA probes, okay, and non-nucleic acid probe, like there are protein probes are also there. So now first into the homologous probes. Homologous probes are nothing but that is having exact complementarity with the nucleic acid sequence which we are searching. Okay, so that is homologous. The non-homologous are nothing but which have similar but not exact complementary sequences are called as non-homologous probes. So for this base pairing to take place, at least 40% homology is going to be always required. So types of nucleic acid probes: oligonucleotide, RNA, single-stranded DNA, duplex DNA is also going to be used as probes. So single-stranded DNA, the most probes are single-stranded nucleic acid fragments complementary to the gene being sought. There is even called as degenerate probes. That's a mixture of all the oligonucleotides that can encode a selected portion of the peptide sequence. Then oligonucleotide probes, which are synthetic single-strand DNAs, RNA probes, which are either it can be isolated mRNAs, fragment can be used, or even we can use the mRNAs directly as probes also. Then we have non-nucleic acid probes, that is antibodies, uh, used for expression libraries and even fusion proteins isolation. People go for not antibodies. There is one more which I have not mentioned here, is the DNA binding proteins. That is also people use it as probes. So this is just what do you call it as a map, which is a representation of these probes and how they are being used. And all these probes, whenever we are going to use, we have to identify the hybridization or complementary pairing of those particular probes, or hybridization with the respective binding with the respective sequence. We need to see it, to visualize it we need to label them. There are different labeling people do labeling with radioactive labeling, and even they'll go for fluorescent molecules are also going to be used for labeling. And rarely people also go for enzyme for labeling, which can convert non-chromogenic substance into a chromogenic, that is into a colored substance. Sources of probes: there are the heterologous probes from one another species. We can means if we are identifying a gene in one species, we can take the sequence of nucleotides unknown from the other species, which can act as heterologous. There is even cDNA probes. You can isolate mRNA molecule. From there, you can produce a cDNA that can be used as a probe. So there are even probes based on protein sequences, wherein people use synthetic oligonucleotide synthetic method, where the using the redundancy of genetic code, they are going to produce the nucleic acid probes. For example, for 6 to 7 amino acid sequence, amino acids, if it is known, which is in sequence in a protein. So by using the redundancy of genetic code, we can produce an oligonucleotide which is having 18 to 21 bases, which is going to have a complementarity with the DNA or the gene. Now, specificity, that's a very important aspect of the probes, because they need to go and specifically interact with the target DNA only. The probe must be specific for the gene of interest and should not annihilate with any other part of the DNA or RNA. So then only we can able to use it as a probe. So this is the most purpose of oligonucleotide containing 20 nucleotides is chosen, because when you take 20, so the nucleotide sequence is going to be, it will appear, for example, almost 10 to 12 times within that, it is easy to go for binding. Since all the genome are much similar, approximately 3 into 10 power 9 nucleotides for humans, so 20 nucleotide sequence in a genome usually occurs only one time. So there will be high specificity we can get. That's why 20 nucleotide probes are going to be more preferred. Then how to detect the probes? As I already told you, it should be either radio labeled or it can have non-radioactive labeling. Radioactive labeling, people use P32, H30, H, that is H3, that is tritium. They go for I-25 as a radioactive labeling. In case of non-radioactive, they can go for chemiluminescence. That is, some luminescent molecules are going to be attached, or some flash of light can come after the reaction. Then some chromogenic substances or even fluorescence can be used, which is used in fish, fluorescent in situ hybridization. Now then, how to detect the hybridization? That's what the colored probes are going to give the flash of color in case of fluorescence, or if they are going to have radioactivity, they give radiation. So which can be identified within that particular presence can be identified after electrophoresis. We can subject it to our autoradiography, and then that will give you the presence of the probe, means hybridization is going to be detected. Next, about the probes, degenerate probes. In the absence of a particular gene sequence, a known gene sequence, people go for this degenerate probes where oligonucleotides are synthesized. So that are being used as a probe, which I already spoke to you as how they are being created and used. One second. Okay, here you can see how these particular probes are being prepared by using the amino acid sequence is getting converted into nucleic acid sequence, and then synthesizing those nucleic acid sequence such of 20 nucleotides such a way that they go and hybridize specifically with the complementary sequences, which actually act as a coding strand. Okay, so now what is the use of probes? To search the specific DNA sequences in the library, southern northern blotting or hybridization, people use it. In diagnosis of genetic disorders, people are using in sickle cell anemia, thalassemia, and all these cases. Probes get involved in the formation of a heteroduplex with the template DNA, and is the key for your usefulness of the hybridization. Now, if you come to some of the methods of hybridization, first is colony hybridization. So, if you remember about replica plating, which I discussed earlier, so in the colony hybridization, what people do is here you can make out they prepare a replica plate of the master plate. Then on the replica plate, they are going to put a nitrocellulose membrane. So when the nitrocellulose membrane is spread onto the plate, so the colonies will get adhered to that, and then later people remove that nitrocellulose membrane. So once it is removed, the bacterial cells are going to be still adhered to the nitrocellulose membrane at the specific positions. Then they lyse these particular bacterial cells by using 0.5 normal NaOH. Then they neutralize by using some neutralization buffer Followed by that, they use proteinases so that the bacterial cells can be, the lysed proteins which are left out, they can be digested off. Then they wash it and they bake it at the 80 degrees centigrade, so that after washing, whatever is left is most of nucleic acids from those bacterial cells at the same positions. So then when they bake it, these particular nucleic acids, they get firmly adhered to the nitrocellulose membrane. After getting this particular nitrocellulose membrane, now we call it as a DNA print of those particular colony bacteria that can be subjected for hybridization with the P32 labeled probes for the specific target DNA sequence. So if the presence of target DNA is there in any of those particular colonies, only to that particular position of the colony hybridization will happen. When we do autoradiography, we can get the spot at that position. So now by matching the position of that particular nitrocellulose membrane with the master plate, we can identify the colony carrying the our recombinant DNA vector. So that can be isolated separately and can be further used. So because here bacterial cells are directly being used, the colonies are directly being used for hybridization, it is called colony hybridization. Next is plaque lift method. So if it is not in the form of bacterial colony, instead if it is going to be in the form of plaques, so now these particular plaques will carry the bacteriophage, that is the recombinant bacteriophage. So how this is done? Let's say, so we have developed a recombinant DNA vector, and that vector has been used to that is a recombinant DNA phage vector has allowed to infect a bacterial lawn. So after infecting the bacterial lawn, it creates some plaques. For about 6 to 8 hours if you incubate, small plaques are going to be created. If overnight means large plaques are going to be created, so which is going to be more likely desired. So then people take that particular plates and they cool it to 4 degrees centigrade so that the agar is going to become more stiff. Then above it they place the nitrocellulose membrane and it will be allowed to be there for 30 seconds to 2 minutes. What happens now, this particular nitrocellulose membrane will get attached with the bacteriophages which are there in those particular plaque region. Then they take that particular nitrocellulose membrane, the plate is going to be kept as it is, as a master plate. Now that membrane which is attached to the phage particle will be subjected for um, it will be first soaked with the alkali so that DNA can be denatured, then it is neutralized, followed by that it is going to be heat fixed by baking at 80 degrees centigrade. Then they are going to add the probes for hybridization, and after adding of probes, then it will be washed off. Unbound probes or unhybridized probes are going to be removed. Then autoradiograph of that particular nitrocellulose membrane will be done. So now wherever we find the patch in the radiogram, so that indicates there the radioactivity has come because of the presence of the vector, recombinant DNA vector carrying our target DNA. Now those particular positions can be compared with the master plate, and the plaques present in those positions will carry our recombinant DNA. So we can actually take out the sample from there. That means these are the bacteriophages which have our recombinant DNA. So this is called plaque lift method because we first lift the plaques with the nitrocellulose membrane and then we are going to go for hybridization, because generally all hybridizations are done here on the nitrocellulose membrane. Okay, so the next one is the immunological screening method. So it's called, you can see immunological. So in this slide I have put two pics. Immunological because here it is, we are using the antibody reactions. Okay, that is a chemical reactions for screening purpose. So here you can see we have placed a lambda, a lambda zap one of the vector system is being used carrying a LAGZ gene with a polylinked DNA. Now when this particular lambda phage with the recombinant DNA is going to infect, or when it is going to be allowed to transform into an E. coli, so it is going to cause plaques, and then they go for plaque lift method so that they lift up those particular plaques by using nitrocellulose membrane. After lifting that particular plaques, then those nitrocellulose membrane are carefully removed and it will be absorbed, because this particular nitrocellulose membrane will be attached with the fusion proteins which have been created as a lambda fusion, sorry, so which have been created as a lambda fusion, LAGZ fusion proteins within the lambda zap. So now this protein, when they get produced, they'll have lambda LAGZ portion as well as our target DNA portions. So as we know, we will be having an idea about what is the gene product of our what is our target gene product. We can develop monoclonal antibodies against these particular gene product, and even these antibodies can be made attached with an enzyme, or these particular antibodies can be labeled either with radioactivity or fluorescent, or even we can go for labeling with the enzyme which can convert a non-chromogen into a chromogen. So now when these antibodies are added to this nitrocellulose membrane, they specifically goes and binds only to that particular region of the plaque, means that particular plaque on the are on the portion of the nitrocellulose membrane which carries the fusion protein. So we can incubate for some time and then wash off excess of antibodies. Then later that particular membrane can be subjected for, that nitrocellulose membrane can be used by for chemical reaction, either for autoradiography or for enzyme labeled means non-chromogenic into chromogenic means we can use specific substrates which forms a color at that particular position, or we can use fluorescence if it is coming at that particular position, which can be compared with the master plate, and we can pick the positive plaques, and those particular positive plaques can be retained. So the other same thing which has been shown here by using autoradiography with I-25. Okay, so this is what the immunochemical screening method. It's one of the method for screening for the proteins. Okay, so that is in case of expression vectors. If the recombinant DNA vector is an expression vector. Earlier nucleic acid hybridization is for the one without expression, means even though if we are not actually looking for expression also, that is for the synthesis of protein, we can able to screen. So in the immunochemical method, in case if we are looking for the protein synthesis, then you can say screen based on the protein synthesized out of our target DNA. So these are few methods of screening. I'll stop here. Again further I'll continue with the blotting techniques later. Okay, so thank you.